Okay, so it's my great pleasure to welcome Maxie Roessler to this LCN seminar. So my name is Sandrine Hoytz. I'm the LCN co-director. So it's really a great pleasure to have you here, Maxie. So Maxie is a senior lecturer in the Department of Chemistry, which she joined in April 2019. She's been very productive since she joined, having set up the 2.3 million EPSRC strategic equipment grant, establishing the Center for Post EPR Spectroscopy at Imperial called Pepper, and a great addition to the capabilities of Imperial, and which had its launch in September as one of the first live in-person events to take place. Maxie has completed her DPhil in EPR spectroscopy on iron sulfur clusters in enzymes at Oxford, completed in 2012, and then moved on to Queen Mary, for her independent academic career in 2013. So we are really looking forward to hearing your talk. So the floor is yours. I think we are recording. Hopefully this is all okay. I should also point out to previous LCN seminars that were centered on facilities, and that Alberto, who's on the call here as well, presented. So there should be also further details on Pepper that can be looked at as part of the LCN seminar series. So yeah, the floor is yours, Maxie. So we'll everyone, I think, is muted, and we'll leave questions for after or in the chat. And thank you very much.
Yeah, great. Thank you so much, Sandrine. And I think maybe up front here, since you mentioned, bringing Pepper here, I think it's fair to say you played a big role in this, Pepper, being here at Imperial. So thank you, Sandrine, here in front of the whole audience. I was going to mention that at the end, but I might as well do that now. So there you go. Let me just start sharing my screen. Okay.
I should also add, sorry, I didn't get my notes straight. Also, that as testament to Maxie's involvement in the EPR community, she's currently the vice president of the International EPR ESR spectroscopy. So great to have this international reach as well.
Thanks, Sandrine. Yeah, by all means, if you have any questions about the IES, I'm very happy to answer those as well. Okay, so I thought I'd start by giving you a little bit of an overview to what we do in the group. So we combine EPR spectroscopy and electrochemistry and biochemistry to unravel the mechanisms of what are some fairly complex redox active enzymes. At least that's a lot the core of what we do. And numerous enzymes rely on redox reactions and are involved in essential biological processes, including respiration and photosynthesis. And luckily, for us at least, these unpaired electrons are often found in mechanistically key locations. And of course, EPR is a really rather excellent method to probe these unpaired electrons. And besides looking at the enzymes in their isolated form, we are also devoting quite a bit of effort to studying them in their native or mimicked membrane environments as of recent, although I won't be speaking about that today. And we are also working on developing a new technique here to study redox-based catalytic reactions through film electrochemical EPR. And that's perhaps the most materials-oriented aspect of our research at the moment. And I'll touch on that towards the end of my talk. And it's very exciting, actually, to be able to dive a bit more into materials chemistry as part of this work. And last but not least, we've got quite a few collaborative projects as part of Pepper, which again, I won't have time to speak about today, but perhaps there's something for the future there. But on the core, I guess, we are focused on looking at enzymes, so looking at biomaterials which have unpaired electrons, but branching out into various directions from there. So in terms of an outline, this is a relatively short talk, so I thought I better focus on something to be able to tell a coherent story. And so I'd like to tell you about our recent adventure into studying photosynthetic complex 1. So I'll give you a bit of context in comparison with a perhaps better known enzyme in respiration that we've been studying for quite some time. And then I'll give you a bit of a background to the post-DPR technique that we've primarily used here for that particular challenge. And that I think will be more applicable to all sorts of systems, so not just that particular biological system we're looking at, so hopefully more generally useful to you as well. And then, of course, I'd like to tell you what we've actually done with that in terms of the characterization and assignment of the iron sulfur clusters in this enzyme. And actually, this brings me back to some of my PhD work that I had kind of left aside for a long time, but it sort of came back in very handy here to really pick up a completely new challenge here in the context of this much less well understood enzyme than the respiratory counterpart. And then for the second part, or well, the last few minutes of my talk, I'd like to give a bit of an overview of pepper, perhaps more in terms of the ongoing work of the unique aspects of it. Of course, Alberto already spoke a few weeks ago, as Sandrine mentioned, and that's perhaps also a place to look if you'd like to know more about pepper. So this is an overview of, I guess, the photosynthetic reaction. So perhaps what most of you are used to in the light reactions of photosynthesis, the electrons are extracted from water and reduce the quinone pool, which is coupled to proton pumping across this membrane, this energy transducing enzyme, and also the reduction of ferredoxin. I should probably try to find a pointer. Let's see if that works. Can you see the cursor? Sorry, can someone?
Yes.
Yes, okay. Then that will probably do. It's not really a laser pointer, but that will do. So we've got the ferredoxin here, and that in turn results in NADPH production for carbon fixation. But what perhaps people are a little bit less familiar with is what happens during, oh, it doesn't want to advance. Here we are. What happens during the dark reactions of photosynthesis. So what can actually happen is that these electrons from ferredoxin go back into the plastoquinone pool through this enzyme here, now known as photosynthetic complex 1. It's not photosystem 1, by the way. I guess often people mix them up, and I think you can be forgiven for that because it's quite a similar name. So it used to be known actually as the NADH-like complex, very much by analogy with NADH ubiquinone oxidoreductase, which is the corresponding enzyme in respiration. But because the substrate is not actually NADH, but ferredoxin, so I'll come back to that, that's really why now in the community it's more accepted to be called photosynthetic complex 1. So what this enzyme does is basically to power up more ATP by this increased proton gradient. And that's thought to be important for plants and also the cyanobacteria that we've been studying in terms of bringing extra energy, if you like, under stress conditions and to promote survival under stress conditions, such as for cyanobacteria under low carbon dioxide concentrations, for example. So this is the sort of long-term angle and motivation for looking at this enzyme, because eventually it may very well be useful to have this basic mechanistic understanding to perhaps engineer more resilient crops or whatever that might be. That's a long way off, but still to give you a little bit of a perspective. And the student who's been driving this is Catherine Richardson. She's submitted her thesis before Christmas, awaiting her viva, and a joint student together with Guy Hanke at Queen Mary. And I think she's done a fantastic job kicking this all off the ground. So this is really all her work that I'm presenting here. So I thought I'd give a little overview of the photosynthetic enzyme versus the respiratory enzyme. So the photosynthetic one's on the left, the respiratory one's on the right. And you can see there's really a very striking structural similarity, which is, of course, how it was first named the NADH-like complex, given this striking similarity. Although there's a big chunk of this hydrophilic domain missing here, clearly, in the photosynthetic enzyme. And there's a few key differences nonetheless. So clearly here we only have 3 iron sulfur clusters as opposed to this really long chain of 8 or 9 iron sulfur clusters. It depends on the species and the respiratory enzyme. But then you've got this big membrane domain here, which is where the protons get pumped through to the other side, which is then used towards ATP synthesis by ATP synthase, where they flow back down their concentration gradient. And unlike the respiratory enzyme, so this is an enzyme that was actually studied by EPR very much long before there was any kind of structural data out there. So it's got a long history of EPR, still nonetheless lots of questions to answer, and that's one of the things that we are doing in my group. But that's really very different to the photosynthetic enzyme, where when we started on this work, there was really nothing. There was some very beautiful more physiology work and genetics work, but really nothing in terms of biophysical studies. So we thought, well, was this what we'd acquired over the years in terms of methodology and I guess expertise in looking at the respiratory enzyme, we thought that that was perhaps a good enzyme to tackle together with the biological expertise from Guy Hanke's group. So unlike the respiratory enzyme, for example, we knew nothing about the reduction potentials and how that all fits in terms of the bioenergetics of this enzyme in photosynthesis. Oh, so basically that was our starting premise here. Can we actually determine the magnetic parameters of these iron sulfur clusters? And I'll come back to that, but that's really the first step to even be able to do more interesting things with it, such as, of course, using pulsed EPR, but then also determining their reduction potentials and actually assigning these to the structure. So first things first, these are the continuous wave EPR spectra, which is perhaps the one technique that people would typically come across for EPR. And we looked at 2 species of cyanobacteria. So all the credit there really goes to Catherine and Guy for being able to isolate these in the first place, which is quite a big challenge to begin with. But what we noticed from the spectroscopy side is, first of all, that one of the signals is very much similar to what we saw in the respiratory enzyme, so the so-called N2 signal. That's just the historical name of it. And by the way, I should also say that these iron sulfur clusters, they are actually EPR silent in the oxidized form, the 4 iron 4 sulfur clusters, and they become EPR active when they become reduced. So from that, I guess we deduced that this must be the cluster N2, but the other 2 clusters, that was much less clear. We couldn't really make any firm conclusions as to how we could assign those based on the homology with the respiratory enzyme. And also, I think you'd be forgiven for being very skeptical about this last cluster being there at all, because it's really barely visible in the continuous wave EPR, and the certainty from all that came really from the pulsed EPR studies that we then subsequently did. So on the basis of this, can we actually define the G values? So for those of you who are not familiar with G values, that's basically the equivalent of the chemical shift in NMR for EPR, and also the G strains, basically a further characteristic of these clusters. And can we also assign which of these signals belongs to which cluster structurally? So to do that, to find the G values and the G strains, Catherine basically did a big pile of different types of post-EPR measurements that are based on relaxation times, so T1 and T2 type relaxations, and you can actually exploit the fact that they relax with different times to effectively filter out one cluster and preferentially get another cluster. You have to be very careful when you do that, because overall they're still relatively similar magnetic entities, and therefore they have relatively similar magnetic properties, so it's not so easy to detangle them. But nonetheless, what we could do is to find out that N2 has a relatively long T1 and T2, and 1 has a short T1 and T2, and N0 has a really quite unusual combination of having a relatively long T1 and a short T2. So T1 being spin lattice relaxation and T2 being spin-spin relaxation, loosely defined anyway. So that already gave us perhaps a hint as to where the N0 cluster might be. By the way, we named them that just to avoid too much confusion with the respiratory homologs. So we gave them the names N1 and N0. And as a result, in any case, you can see there's the fittings with the red lines on top. And I guess fitting is quite important here to be able to get to a consistent set of data. And here's the corresponding set of G values and G strains for these 2 sets of cyanobacteria that we investigated, which is pretty consistent between the 2 species, which is reassuring. And here's the comparison with the respiratory enzyme. So on top, you've got the photosynthetic complex, and on bottom, the respiratory one. And again, you can see the pink one here, that's the N2 cluster. That's the corresponding chain in the respiratory enzyme, and they match up very well on the whole. But clearly, N1 and N2 don't really match up with any of the clusters in the respiratory enzyme. And also, I should say that in most complex one, respiratory complex one, actually the cluster that's adjacent to this terminal cluster N2 is EPR silent, even in the reduced form. So the next comparison is really the N4 cluster, which is the next one along. But none of the two G values that we see here in the photosynthetic enzyme matched up with N4. And perhaps it was an interesting discovery in the first place, actually, that we see all three iron sulfur clusters or at least some extent of it, as opposed to the respiratory enzyme, where basically if we truncated it after those three clusters, we would only see two. So basically the question for us to answer from here was to find out which one is the cluster that's adjacent to N2. And to do that, we used dipolar spectroscopy, but basically there's only two possibilities, which somehow makes things a bit simpler, which is basically to either have N0 in the middle or at the end. And to be able to solve this puzzle, we wanted to use double electron-electron resonance spectroscopy, which I think is a technique that Alberto already introduced in his talk, in case you happen to be able to attend that. And the premise here is somewhat different. It's actually not really determining the distances ab initio, but rather using this information that we have from the structure to simulate the data for the two possible models and then to compare them to the experimental data to basically decide which model is correct. So this is what we did, and I thought I'd introduce you a little bit to the DEAR experiment. I won't go into too much detail, but I think it's quite an intuitive experiment to understand, because you can effectively treat the two spins as little bar magnets, and how strongly they interact with each other in terms of the dipolar interaction, as you can imagine, is very much dependent on the distance. And the most commonly used experiment here is still the 4-pole sequence. So you can see here at the top, that's the so-called observer spin sequence. So this is the standard Hahn echo sequence, where we've got the echo here appearing at time 2 to 1. And then if you apply another pi pulse, then you'd simply normally refocus that echo at 2 times tau 2. But when you then apply a pi pulse at a different frequency, so the B spins, then that would effectively interrupt that echo, and then the modulation of that echo is proportional, or well, inversely proportional to the distance cubed between the A and the B spins, and therefore tells us something about the dipolar interaction. And actually, I had a little animation here for you to show you that, what the effect of the B spins is in terms of the effect of the A spins, but you can imagine how that might happen, hopefully. So this is the sort of trace that we see in a DEAR experiment. And this dipolar frequency here, or the period, then tells us something about the distance here, r cubed, between the A and the B spins. I also wanted to tell you a little bit more about DEAR with iron sulfur clusters, because it's somewhat quite unusual magnetic entities. They are very delocalized. So you have to take that into account. It's really quite different to an organic radical. And that has its advantages. It can make the analysis more complicated, but in our case, it also helped us because we have these really broad spectra, so very wide spectra, which means that with a single microwave pulse, we can't excite the entire spectrum. And therefore, we actually get some orientational information depending on where we excite along the spectrum. And indeed, actually, we have to acquire DEAR spectra at many different field positions in order to really obtain this whole picture. But actually, that's what very much helped us with the assignment. And then the spin delocalization, I won't go into too much detail here, but you can take that into account with so-called spin projection factors, which is how much spin you have on each of the ion subsites in this cluster. And last but not least, we also have to worry about which distances contribute. So in principle, there's of course 3 distances, but somewhat luckily for us, actually these shorter distances are very difficult to pick up with DEAR spectroscopy, especially with these sort of delocalized metal centers. So although we looked at definitely simulating these shorter distances as well, we only really have to consider the dominant contributing distance, which is 26 Armstrong between the most extreme clusters. And the modulation dApps here actually informs on the fraction of the B spins, so that are excited at the second microwave frequency, and that has a very strong effect on the DEAR traces. And actually, that's a very good indicator in terms of how good your fit is, whether you're able to match this modulation dApps. So that's certainly something that we also paid attention to. And last but not least, we were definitely facing quite a challenge in terms of sensitivity here. So this protein is really hard to come by, and Catherine had to work very hard to even get very tiny amounts of it. And luckily here we could actually make use of some of the instrument development that's happening in John Morton's lab, and I'll come back to that at the end of my talk here as part of Pepper. So DIA basically enabled the assignment of N0 and N1. So you can see in black is the experimental data, and on top of that you see the simulated data. So in red for model A and in blue for model B. And even though the fit is certainly not perfect for model B, I think clearly it's much, much better than for model A. So for model A, I think it's fairly meaningless to speak of a fit at all. So we could really quite confidently conclude that it must be model B that's correct. So therefore we have the N0 cluster that's the furthest away from N2. And really what gave us the confidence in this assignment as well is these outer edges that I mentioned, that because of these broad field sweeps that we can't excite the entire spectrum, that really helped us with the assignment here. So the fact that we can't excite the entire spectrum. So this enabled us to assign what different spectroscopic signatures actually are with respect to the structurally defined clusters. But you might say, okay, so what? What's that useful for? So that really formed the basis for us then to look at the ein-sulfur cluster properties and in particular at their reduction potentials. And for that, we used some methodology that, again, we developed for the respiratory enzymes, so small volume potentiometric titrations. So potentiometric titrations are extremely well established in the field and have been used for decades, and I think have definitely proven their worthwhileness, but they can be a bottleneck in terms of how much enzyme or how much material they require. So we effectively miniaturized the setup where we need at least an order of magnitude less sample, which really made it feasible at all to do this. Because there's absolutely no way that we could have got several milliliters of this enzyme to do it. So to cut a long story short, Catherine did these titrations. So basically what you do is just to have your pot of enzyme and solution, and you also add some mediators to communicate the potential of this large buried redox center to the working electrode. And then you adjust you add chemical reductants and oxidants to adjust the potential, and then you read it out with an open circuit potential. So here we have the reduction potentials of clusters N1 and N2, so the purple and the pink one. And much to our surprise, actually, they were basically isopotential, so the same same potential for both. That's not really something we were expecting, to have two adjacent isopotential clusters, because normally that would be considered as not being very energetically favorable. And in fact, lots of these sort of electron transfer relays are thought to be roller coaster type energy profiles, but this does not seem to be the case here. So perhaps that's mechanistically relevant. And that's, of course, something that we are investigating further at the moment. Perhaps to do with the fact that actually the enzyme takes a single electron donor as opposed to a double electron donor in the respiratory enzyme, and then still picking up 2 electrons to reduce the ubiquinone. Sorry, I should show that on the next slide to make that clearer. So I should also say that at this point, we can't actually assign the reduction potential of N0 because this was not easy to pick up in the continuous wave EPR, as I mentioned at the beginning. It is there in the post EPR, but we could at least put a bound on it. So we know that it really has to have quite a negative potential, lower than half a volt or so, which is really quite negative. So that brings me to conclude the most part of my talk. I just want to say a few words about Pepper afterwards. So even though it's just the beginning, at least I think we've managed to place this enzyme on the bioenergetic map of photosynthesis. Clearly, there's lots more work for us to do, even some very basic questions like developing a proper activity assay for it. And Catherine had some promising first leads in that. But really what I hope I've shown you here is that we could use a dipolar technique here, so double electron spectroscopy, so a pulse DPR technique to single out these relatively, I guess, weak dipolar interactions from all these other spin Hamiltonian parameters, and that certainly we can only do with pulse DPR. And hopefully I've given you a bit of a flavor as to what you can do with pulse DPR, and that's also certainly applicable to completely different systems. So I guess one of the powers of pulse DPR, right, that you can really single out different types of magnetic interactions that you wouldn't be able to pick up in a continuous wave EPR experiment. And we have, of course, determined the redox properties of the cluster, or at least 2 of them. So that's something that we still need to scratch our head over as to what it means. It could prevent unspecific electron transfer back to the ferredoxin. Perhaps it also helps in terms of preventing ROS production. It could be that these 2 clusters adjacent to each other are easily holding an electron each, ready for the plastoquinone, which is a 2-electron reduction, of course, to pick up these electrons more efficiently, but that's really all speculation at this stage. And of course, for me, it's interesting to see as well what are the differences with the respiratory enzyme, and can we actually eventually figure out what this energy coupling mechanism, as we call it, so the way that the electrons are used, so the free energy is used to pump these protons against their concentration gradient to make ATP. Can we figure that out, or can we contribute to that at least? So then I'd just like to spend a few minutes to tell you a bit more about Pepper. So I like to think of Pepper as being built on 3 pillars. So the first yellow one here I've already told you a bit about, so the post-DPR aspect. I didn't tell you much about the multi-frequency. So we actually, all these experiments that I spoke about were done at X-band, the most common frequency, but we also have Q-band, which is quite exciting, and especially for distance measurements can be really very much an advantage. But there's also 2 other pillars here, so the orange on the and the gray one that I'd like to tell you a bit more about. And of course, the first one, the post-DPR, very much spoke about the protein function aspect, which, by the way, these sort of 4 categories that we identified for the grant are areas of particular strengths that we felt we could make a case to the EPSRC for. And there's really exciting work going on across Imperial, and of course also in Sandrine's lab, more on the materials side in these different aspects here. But in terms of the new capabilities that we can hopefully bring to the field of EPR, so let me tell you a little bit more about that. So the first one here is a technique that we are developing in my group, and that we've coined film electrochemical EPR, and it was actually kicked off by my former student Kaltum. And what we want to do here is to combine EPR spectroscopy with film electrochemistry. And film electrochemistry is basically like electrochemistry, except that we are sticking a redox active species directly on the working electrode, which has a lot of advantages, primarily because we are eliminating the diffusion problem, which is a big problem when you have big enzymes like what we are looking at. And what we've already shown is that we can achieve direct and accurate potential control of buried redox centers in an enzyme, which is non-trivial because normally they are just too large to be directly controlled like that. That's why we need these mediators in solution. And well, why do we don't want to do that? Because ultimately, what our dream is, if you like, is to be able to really watch radicals evolve in operando during catalysis, during enzyme catalysis, but in principle that can be any redox active species. And we haven't published that yet, but on the right here you can see that we are getting towards that to really watch the radicals evolve during a reaction here. That's for a tempo derivative, so for a nitroxide type derivative. And here's my one more materialsy slide. So at the moment, the 2 people driving that further is Mariam and Yunfei from my group. And we're looking at different working electrode materials here. So on the left is ITO-based materials, so indium timoxide. And this is in collaboration with Erwin Reisner's group in Cambridge. And really the possibility to tune these structures into different hierarchical structures to suit different purposes. And on the right here is a more recent adventure. This is together with Milo Schaffer here at Imperial. And Yunfei really drove that forward very efficiently in his MRES project last year to work with CNT-based materials for the working electrode, which we hope has other advantages, such as extending the potential range that we are able to look at compared to the indium tinoxide. So in terms of the second, or the third pillar, so the instrument development aspect, I should say up front here that this is very much driven by John Morton at UCL, who is also part of the LCN, of course, and his former postdoc Mantas, who's now setting up his own lab in Lithuania. And so our contribution here was very much, I guess, to show a real sample contribution to what is, I guess, a method development in the first instance. And this setup effectively uses cryogenic preamplifiers. And what we've shown here is that we can enhance the signal to noise by an order of magnitude with these cryoamps. And that's really a huge impact in terms of what we are able to do spectroscopically. And I didn't really spell that out very clearly so far, but lots of these pulse EPR measurements, even though they can be very powerful, they can also be very long and slow. So sensitivity can be a huge bottleneck. So it's something that is more generally applicable, I think, to the whole field. So what we've shown here in the context of the respiratory enzyme is looking at the proton couplings that are close to this terminal iron sulfur cluster N2, and you can see the difference in the measurement or in the quality. They're both acquired for an hour. So with the conventional setup on the right-hand side, you really don't see anything at all, whereas with the cryo-amps, you really get quite a nice set of signals after just an hour. And this is definitely applicable to all sorts of EPR techniques, including the spectroscopy that I spoke about. Here it's applied to more conventional spin labels, so nitroxide type spin labels. And you can see how in the same measurement time here with the conventional setup, it's really quite lousy. The signal to noise was a very uncertain distance distribution as opposed to the setup with the cryo-amp. So going forward, I think there's lots of exciting directions for us, not just to make measurement acquisitions faster, but also to be able to probe spin setup systems that we wouldn't be able to probe at all in the absence of having this sensitivity enhancement. Such as surface spins, near surface spins. So I hope that will be exciting also more in the materials applications. But also we are exploring completely new directions as well with microresonators, which I don't have the time to speak about here, but which I'm happy that John and I now got some funding to push this project forward as well. And then very last but not at all least, I'd like to thank the people from my group who've been involved in these projects. And I think I've mentioned them along the way, but also very much our collaborators. Of course, Guy was very much involved in the photosynthetic work, and John, I mentioned on the instrument development, various other people in contributing to this project. And last but not least, thank you for everyone who's been involved in building up Pepper, and first of all, getting the funding and then actually making it happen. And that's, of course, very much also Alberto, who I think is in the audience. And Sandrine, I mentioned you at the beginning already, but all your support from the beginning has really been so important for me. And thank you all for listening. And I hope I didn't take too much time at the end here and with the interruption. And of course, I'll be very happy to take any questions.
Thank you so much, Maxie, for this wonderful talk. Just combining, detailed project and then the broader perspective of Pepper. That's really excellent. So the floor is open for questions. If anyone, please just unmute your mic or type it in the chat. I'll just kick off on the high sensitivity setup that you are developing with John. What is the minimum number of spins or equivalent film thickness or so on that we could be working with in practice? I know that for the audience that might be useful to.
Yeah, I think I'm not sure I can give you complete absolute numbers at this stage, Sandrine. I think when we get on to moving with microresonators, that will further come down. I mean, typically, I suppose what you would say, and I mean, Alberto, feel free to chip in here as well, I think is 10 to the 13 spins or so to get a signal for continuous wave EPR. If you have a very narrow signal, maybe 10 to the 11. And I guess in principle, if you go down by an order of magnitude, then you can go, well, another 100 down or so from that, but it really very much depends on how thin or how wide your line is, your EPR line, because of this anisotropy that I spoke about. And it also does depend a bit on the sort of experiment that you're looking to carry out. But we can certainly try and do a back of the envelope calculation to work out if you really had a thin film as to what the number of absolute spins would be that are feasible to study with this. But moving forward, I think the microresonators will also be a very exciting direction with that, especially for looking at surfaces.
Okay. Thank you very much. Any further questions?
I think there was a question from James. Does your workflow also work with proteins, i.e. cryo? James, maybe do you want to ask your question, if that's okay? I'm not quite sure what you mean.
Oh.
Maybe he's left. No. Ah, okay. Was that the
yeah,
presentation? Maybe.
Yeah, I'm not seeing it.
It says thanks both for it. Okay, James, I guess there was a previous talk, maybe I missed it. I don't know. Sorry about that if that's the case.
On the distance measurements, what is the accuracy that you can have on these, and also how does it cope with the spread of distances when you're doing DA measurements?
Yeah, so I mean, I guess when you use the more commonly used spin labels, they can be quite conformationally labile, and then you get definitely broader distant distributions. So I guess the more conformational freedom you have, the broader your distance distribution would be. I mean, in terms of accuracy, I suppose in our case, we kind of knew what the distance was to begin with, right? So I guess we were trying to simulate the corresponding models and see which one would be correct. So I guess in a way, the accuracy is less important for us there. But I mean, generally, it's, I guess it's pretty accurate. It depends again on what it is that you're looking at, whether it's very delocalized magnets, but say you have a distance of, I don't know, 26 angstroms with a nitroxide, then I guess you could definitely say plus or minus a couple of angstroms at most, I would say. I don't know, Alberto, if you want to comment, and feel free. You know more about spin labels in DIA than I do.
It's been put on the spot, Alberto.
No, yeah. Anyway, but because I guess we've used it in a more unusual context like this, in terms of these delocalized metal clusters. But yeah, I mean, I think on the whole, it's a very well-established technique that I think you can rely on if you do it properly and you analyze it properly.
Yeah, I'm thinking, dopants within semiconductors and a distribution of nearest neighbors. So when it's a bit more of a statistical distribution.
Okay, I see. Yeah, no, then it can get tricky, I suppose, if you don't have a well-defined distance, but you've got lots of ones. I mean, there's now work, of course, looking at also 3 spin centers, even 4 spin centers. Then it can get quite tricky if you've got multiple, I guess, sets of dipolar interactions, and then you basically get a complete mix of them. That can be more tricky, definitely.
Yeah, but hopefully doable and of interest. Okay. Anthony, you have your hand up. Go for it.
Yes, I do. Thank you. Yeah, thanks, Maxie, for this very nice talk. I had a curiosity question about the nice ITO electrodes you were mentioning for electrochemical probing. Maybe can you comment a little bit further about that? What is the kind of impact of the porosity, the structuration of the electrodes? Maybe switching at some point, I don't know if it's interesting, from porous ITO towards nanoparticle-based ITO at some point?
Yeah, sure. So, I mean, actually, I didn't really talk about it in any detail, but it is a bottom-up approach. So it is basically starting from, at the moment, commercially available nanoparticles, and then building up these hierarchical structures from that. So in the first instance, just the so-called meso-ITO, but then also using, I guess, in the first instance, Erwin developed it with like basically polystyrene balls that they were burning out. That didn't work for us, so we found various other ways to adapt that. But it is for us, I mean, an interesting system, which is how I first got talking to Erwin, because Erwin Reessler, our collaborator in Cambridge, because it does enable us to basically tune the pore size to what can be quite large proteins. And I think in terms of achieving the sensitivity as well, I mean, ultimately, I think it would be much neater and perhaps more informative for us to be able to really go to monolayers of ITO and to really have single protein molecules just on the surface like that. And I think that's something that hopefully eventually we will be able to do together with the microresonators in John's lab. But at the moment, at least, in order to get that sensitivity, because it's such a compromise to get the conductivity you require for the electrochemistry and the lack of conductivity that you ideally want for the EPR, that's always a compromise at the moment until we move to microresonators, we hope. So basically then these hierarchical 3D structures, they really help, because we basically have lots of protein packed into a very small area, which is what we need in our EPR experiment. But I think there is lots for us to understand still in terms of really how our proteins interact with the surface, how to optimize that process. And actually, this is something I didn't mention. I should have mentioned it, but this is something that we are really quite excited now in terms of tapping into some of these amazing facilities that they are in the materials department. And one of my PhD students has been talking to James O'Douglas, actually, who's definitely posted something in the chat in the past. So I'm very grateful for that. And I think it's very insightful, actually, to see visually what is going on with some of those techniques. So I think there's a lot of scope for us to improve that much more in terms of how we can really optimize electron transfer from the enzyme to the electrode, and in terms of how we can optimally load it, how we can eventually perhaps move to monolayers. So yeah, definitely this is only in a way a crude beginning what we've done so far. I guess we were surprised that we got this good sensitivity at all. And I think at the time when I submitted the Leverhulme Grant Trust on a project on this, one of the reviewers commented like saying, not sure you're ever going to get the sensitivity. So we were surprised that we could get that sensitivity. But yeah, I hope that answers a bit, bit more.
Yeah, definitely amazing.
Yeah.
Thank you.
Yeah, and I know you've done lots of nice work on ITO. So yeah.
Thank you. Yeah, of course. That was interesting. Thank you.
Okay, thank you very much, Maxie. This is normally the time when the sandwiches should be wheeled in the real world. And where we could carry on informal discussions, but hopefully we can do this at some point in not the too distant future. I don't think there are any further questions in the chat. Thank you very much for the presentations. Also, you kept to time and we kept going. So that's all great. And hopefully, if there are further questions, they can carry on in the chat, which is a good repository.
Yeah, very happy to. Hopefully, at some point there will be the opportunity to interact in person, which is always a bit easier, isn't it? So yeah, that would be great.
Okay.
Thank you very much. Yeah, thanks everyone for joining and for listening. Yeah, thanks Sandrine for having me.